So good morning to all of you and welcome back again in our online lectures for Bhalerao Science College Sauner. So dear students, we are discussing here the part of BSc Semester 3 Paper 1 having the name Angiosperm Systematics, Embryology and Indoor Gardening. In this we are discussing the part of Unit 3 which is the part of Embryology, which consists of the pollination, types and significance, Anther, TS of Anther, microsporogenesis, structure of pollen grains, development of male gametophyte, ovule, types of ovule, structure of anatropous ovule, then megasporogenesis, development of female gametophyte, that is a polygonum type of development. And last topic, that is the fertilization, which is the part of double fertilization and a triple fusion, endosperm and its type, structure of dicot embryo, onagrade type, and a monocot embryo. So all these points are involved in this unit number 3. In the previous lecture, we have completed the part of pollination, types of pollination, significances of pollination, and also we have discussed the part of microsporogenesis, that is which is related with the structure of anther. In this structure of anther, which is totally related with the development of male gametophyte, and this male part of the flower, it is called as Androstrum. The Androstrum, it consists of the individual members which are called as stamens, and stamens consist of 3 important parts: anther, filament, and a connective. In the yesterday's lecture, or in the previous lecture, we have discussed the part of structure of anther. This structure of anther, it consists of the 2 lobes or sometimes 1 lobe. 2 lobes, that is a dithecous, 1 lobe monothecous. 2 lobes, transverse section of TS of anther, which produce the tetrasporangiate structure, means it produces 4 pollen sacs or the pollen chamber, or we can also call it as microsporangia. In the previous lecture, we have discussed the diagrammatic representation of the TS of anther also. In these 4 chambers are there, pollen chambers 1, 2, 3, and 4. And after maturity of this anther, it becomes merge. Okay, anther lobes जो होते हैं या फिर जो pollen chambers जो होते हैं वो merge हो जाते हैं. Yesterday we have discussed the microsporogenesis. ठीक है, हम लोगों ने microsporogenesis भी वहाँ पे discuss किया था, and also we have discussed the structure of microspore, that is the structure of pollen grain. So let's just take a look regarding this again microsporogenesis and the structure of microspore. Actually, the part of structure of pollen grains, and later on we will start again the next topic, which is the part of development of female gametophyte, which is started from the structure of ovule. So let's just simply revise this portion by reading the sentences. Structure of microspore. Typical pollen grain is a non-motile haploid unicellular body with single nucleus. It is surrounded by the 2-layered wall called as sporoderm. Outer layer is exine, inner layer is called as intine. Outer layer is made up of the chemical substance, non-biodegradable chemical substance, which is called as sporopollenin. It may be a smooth or with sculptured pattern. It is resistant to the chemicals. At some places, exine is very thin, showing the thin areas known as germ pores. These are meant for the growth emerging pollen tube during the germination of pollen. The inner wall layer is called as intine, consists of the cellulose and the pectin. So this is the structure of pollen grain and the development of male gametophyte. Yesterday we have in the previous lecture we have discussed this. This is the structure of pollen grain, which consists of the exine, intine, cytoplasm, then nucleus. It is divided into the 2 cells, vegetative cell and the generative cell. Again, there is production of germ pore. Then process of cell division, karyokinesis and cytokinesis is carried out, and there is a development of 2 non-motile male gamete. Then there is a formation of pollen tube. So the pollen tube along with structure of pollen grain, this complete structure is called as the male gametophyte. The second mitotic division is concerned with the generative cell only and give rise to the two non-motile male gametes. The mitotic division of generative cell takes place either in a pollen grain or in the pollen tube. The pollen grains are shed off from the anther at this two-cell stage in most of the angiosperm. So let's just talk about the next part, that is the ovule. Let us talk about this ovule. Ovule के बारे में हम लोग यहाँ पे देखते हैं कि ovule it is related with the development of female gametophyte and part of female reproductive system in case of the plants. The female reproductive whorl of the flower is the gynoecium, which is commonly called as a pistil. ठीक है, उसे pistil भी कहा जाता है. The individual members of gynoecium are called as a carpel. ठीक है, अगर individual member अगर single member लेते हैं gynoecium का, then it is called as the carpel. ठीक है, तो कुछ इस तरह से वहाँ पे स्ट्रक्चर मिलेंगे आपको. You will get the structure like this, which consists of again the three portion. That is, you have, I think, studied in the previous lecture, which is the part of this is the stigma, this is the style, so that the female reproductive whorl of the flower, which is called as the gynoecium, and the individual members of that female reproductive whorl is called as the carpel. So this carpel is as like this, which consists of the stigma, style, and the ovary, the part of ovary. So in this way, we can say that the stigma, style, and the ovary is there in case of the carpel. So this is the stigma, this one is the style, and this is the part of ovary. Inside the ovary, there is the presence of structure of ovule. Structure of ovule is there. ठीक है, यहाँ पे ovule का structure मिलेगा आप लोगों को. You will get the structure of ovule, which is basically the anatropous ovule. ठीक है, वो anatropous ovule होता है. The female reproductive whorl of the flower is the gynoecium, which is commonly called as a pistil. Individual member of gynoecium is called as a carpel, that is a megaspore. A flower with many free carpels is called as apocarpus, that is a Michaelia example. And a syncarpus flower is one that has many carpel fused together, for example the brinjal. Typical carpel has a three parts, for example ovary, style, and stigma. जैसे कि हम लोगों ने अभी भी देखा stigma, style, and ovary. तो तीन हिस्से होते हैं एक सिंगल कार्पेल के. The number of ovules in the ovary varies. For example, paddy, then wheat, and mango are the uni-ovulate, whereas the tomato and the lady's finger are multi-ovulate. तो यानी कितने नंबर ऑफ ओव्यूल्स वहां पे प्रेजेंट है उसके बेसिस पे यूनी ओव्यूलेट और मल्टी ओव्यूलेट स्ट्रक्चर ऐसे बनता है, ऐसे इन लोगों ने यहाँ पे बोला है. Now let's just talk about this megasporogenesis. Now what is mean by this megasporogenesis? ये megasporogenesis क्या होता है? It is the process of formation of haploid megaspore. ठीक है, the process of formation of haploid megaspore from the diploid megaspore mother cell, that is MMC, is called as megasporogenesis. इसे हम लोग megasporogenesis कह सकते हैं. ठीक है, so the megasporogenesis, that is the process of formation of haploid megaspore from the diploid megaspore mother cell is called as megasporogenesis. Megaspore mother cell becomes distinguished in the nucellus, more or less in the center but towards the micropylar end of the ovule. ठीक है. मेगास्पोरोजेनेसिस जो होता है it is the process of formation of haploid megaspore. ठीक है, वहाँ पे haploid megaspore बनने की प्रोसेस होती है from the diploid megaspore mother cell, and the megaspore mother cell becomes distinguished in the nucellus, more or less in the center but towards the micropylar end of the ovule. So the megaspore mother cell becomes distinguished in the nucellus. ये nucellus जो होता है वो ovule के अंदर होता है, वहाँ पे megaspore mother cell develop होती है. Megaspore mother cell becomes distinguished in the nucellus, more or less in the center, but toward the micropylar end of the ovule. Now let us talk about this development of female gametophyte, that how this female gametophyte can be developed. Megaspore mother cell undergoes the meiosis to form the linear tetrad of haploid cells, that is the megaspore. ठीक है, मतलब मेगास्पोर मदर सेल जो होती है इट अंडरगोज meiosis. ठीक है, meiosis मतलब सेल डिवीजन का हिस्सा होता है to form the linear tetrad of the haploid cells, that is the megaspores. Upper three megaspores about and lowest one towards the center of the nucellus remains functional. मतलब देखिए चार वहां पे मेगास्पोर मदर सेल बनती है और चार में से तीन जो होती है वो डिजेनरेट हो जाती है. ठीक है, 1 2 3 एंड 4. ठीक है, तो इन चारों में से एक जो होती है वो डिजेनरेट हो जाती है, सॉरी बाकी की तीन जो होती है वो डिजेनरेट हो जाती है और एक बच जाती है. ठीक है, तो एक जो बचती है उसे फंक्शनल मेगास्पोर करके कहा जाता है. ठीक है, अपर थ्री मेगास्पोर अबाउट, ठीक है, ऊपर की तीन जो होती है अबाउट हो जाती है, मतलब डिजेनरेट हो जाती है, and the lowest one toward the center of the nucellus remains functional. It acts as a first cell of the female gametophyte. ठीक है, ये जो सेल रहती है फिर उसे फीमेल gametophyte कहा जाता है, first cell of female gametophyte. Generally, one megaspore toward the center is functional megaspore. मतलब एक मेगास्पोर जो होता है मेगास्पोर टुवर्ड द सेंटर इज फंक्शनल मेगास्पोर. इट इज इन फैक्ट द फर्स्ट सेल ऑफ द फीमेल gametophyte. ठीक है, ये फर्स्ट सेल होती है फीमेल gametophyte की. इट अंडरगोज द थ्री सक्सेसिव फ्री न्यूक्लियर माइटोटिक डिवीजन. ठीक है, मतलब ये जो सेल होती है वो तीन बार न्यूक्लियर डिवीजन और माइटोटिक डिवीजन करती है. Thus, a total 8 nuclei are formed, 4 of which are located at each pole. One nucleus from each pole migrate towards the center and called as a polar nuclei. 3 nuclei toward the micropylar end constitute the egg apparatus. It consists of the large central haploid egg cell and 2 supporting haploid synergid cells. Synergid shows the hair-like projections called as a filiform apparatus, which guide the pollen tube towards the eggs. ठीक है, now next portion that is a part of structure of anatropous ovule. structure of anatropous ovule देखेंगे और फिर से देखेंगे कि किस तरह से यहाँ पे फीमेल gametophyte डेवलप होता है along with the diagrammatic representation also. ठीक है, तो diagrammatic representation भी वहाँ पे लेंगे हम लोग और structure of anatropous ovule भी वहाँ पे अच्छी तरह से पढ़ेंगे. So I think आज के लिए बस इतना ही पढ़ेंगे. I think we have to stop here. Remaining portion we will discuss in the next lecture. तब तक के लिए सब लोगों को stay home, stay safe, सब लोगों को bye. और आप लोगों को attendance देनी है आपके WhatsApp ग्रुप पे.